Videos is on human metabolism, and it won't go into too much detail about glycolysis, gluconeogenesis, beta oxidation, etc. It will just show you how they're all interconnected in some way. And also there are some pictures such as this glucose molecule, which is a 6-ringed carbon structure, to help for visual learning. Let's begin this journey with glycolysis, and again this won't go into detail. I will introduce the products of glycolysis first, then I will go back and introduce the enzyme and other cofactors, ATP, etc. Glucose converts to glucose 6-phosphate with the addition of a phosphate on the 6th carbon. Glucose 6-phosphate will then change its structure to form fructose 6-phosphate. Exactly the same, except it's a 5-ring structure, still with the 6 carbons. Glucose 6-phosphate and fructose 6-phosphate are isomers of each other. This line with the 2 arrows indicate that the reaction can be reversed, whereas the reaction before can't be reversed without using a different enzyme. Fructose 6-phosphate will convert to fructose 1,6-bisphosphate with an addition of a phosphate. So now there's a phosphate on the 1st and the 6th carbon, a phosphate off the 1st and 6th carbon. Fructose 1,6-bisphosphate will then split in half to form 2 structures. One of them is called dihydroxyacetone phosphate, and the other one glyceraldehyde 3-phosphate. Now the difference between the two is that glyceraldehyde 3-phosphate is in an aldehyde form, with the oxygen coming off the end, a double bonded oxygen coming off the end. And the dihydroxyacetone, however, is in a ketone form, with a double bonded oxygen coming out in the middle. Now in glycolysis, dihydroxyacetone phosphate will typically convert glyceraldehyde 3-phosphate to continue glycolysis. But I'll show you this later on. So glyceraldehyde 3-phosphate will continue glycolysis and convert to 1,3-bisphosphoglycerate with an addition of a phosphate to the 1st carbon. 1,3-bisphosphoglycerate will then dephosphorylate to become 3-phosphoglycerate, with the phosphate still remaining on the 3rd carbon, but the phosphate off the 1st carbon has gone. So 3-phosphoglycerate will then convert to 2-phosphoglycerate. The phosphate off the 3rd carbon has just changed positions and is now off the 2nd carbon. So 2-phosphoglycerate will then dehydrate, removal of water to form phosphoenol pyruvate. And fine, this is what phosphoenol pyruvate looks like. And then finally, phosphoenol pyruvate will dephosphorylate to become pyruvate. And dephosphorylation, the removal of the phosphate from off the 2nd carbon. Now pyruvate is the final product of glycolysis. So now let's go back and look at the enzymes and stuff. So glucose with the enzyme hexokinase will get a phosphate group from ATP to form glucose 6-phosphate. And this reaction is irreversible, cannot be reversed unless with a different enzyme. Glucose 6-phosphate, then with the enzyme phosphohexose isomerase, will change shape to fructose 6-phosphate, which is an isomer of glucose 6-phosphate. This reaction is reversible. Fructose 6-phosphate with the enzyme phosphofructokinase will get a phosphate from ATP again and form fructose 1,6-bisphosphate. Now this reaction is reversible. Then fructose 1,6-bisphosphate will split in half with the enzyme aldolase to form glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is reversible. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate can convert back to each other with the enzyme triose phosphate isomerase, because both these molecules are isomers of each other. However, in glycolysis, dihydroxyacetone phosphate will usually convert to glyceraldehyde 3-phosphate to continue glycolysis. So there are 2 glyceraldehyde 3-phosphates now in glycolysis, but for simplicity, I will only pretend there's only one. So 1 glyceraldehyde 3-phosphate with the enzyme glyceraldehyde 3-phosphate dehydrogenase will convert to 1,3-bisphosphoglycerate, the phosphate coming from inorganic phosphate. And also in this reaction, hydrogen was removed by NAD to form NADH. 1,3-bisphosphoglycerate with the enzyme 3-phosphoglycerate kinase will dephosphorylate the phosphate from the 1st carbon by transferring it to ADP, making 3-phosphoglycerate. 3-phosphoglycerate then, with the enzyme phosphoglycerate mutase, will form 2-phosphoglycerate. The phosphate is only in a different location now. It is off the second carbon. 2-phosphoglycerate will dehydrate, remove water, to form phosphoenolpyruvate by the enzyme enolase. And finally, phosphoenolpyruvate will dephosphorylate by the enzyme pyruvate kinase to form pyruvate. The phosphate is transferred to ADP, forming ATP. Pyruvate is the final product of glycolysis, from glucose all the way to 2 pyruvate molecules. Pyruvate looks something like this. Now, glycolysis is a 10-reaction process to provide quick energy in the forms of ATP. So glycolysis is to provide quick energy, such as when running, like this, or sprinting. Remember that 2 ATP was invested or used until fructose 1,6-bisphosphate. Then, from 2 glyceraldehyde 3-phosphates, 4 ATP was produced in total, giving a net of 2 ATP, a net gain of 2 ATP. Now, the 2 pyruvates then have a number of fates from glycolysis. In anaerobic conditions, or without oxygen, it can enter 2 things and become 2 things. One of which, 2 pyruvates, can become 2 ethanols and carbon dioxide, and 2 carbon dioxides. And this is during alcohol fermentation to make wine or whatnot. Or alternatively, the 2 pyruvates can produce 2 lactates, which is a byproduct of muscle when using glycolysis for energy, and occurs when exercising vigorously. Alternatively, the 2 pyruvates can become amino acids, such as alanine, which has a very similar structure to pyruvate. But mostly in a metabolic environment, during aerobic conditions with oxygen, when the body needs more energy, pyruvate will enter the mitochondria and convert to acetyl-CoA. Now, acetyl-CoA can then enter the citric acid cycle, also known as the Krebs cycle, to synthesize more ATP. So the mitochondria here consists of 2 separate membranes, the outer membrane and the inner membrane. Pyruvate enters the mitochondria through the pyruvate transporter, through the outer membrane and the inner membrane. So pyruvate is now inside the mitochondria. And pyruvate will then convert to acetyl-CoA, releasing carbon dioxide, CO2, with a pyruvate dehydrogenase complex, the enzyme. Now, it's a large complex because it consists of 3 enzymes, thiamine pyrophosphate, or TPP, lipoate, or lipoamide, and FAD, which is one of those electron carriers. Now, acetyl-CoA is an important metabolite and interconnects with almost everything. It is also important in fatty acid synthesis, which we will look at later on. But typically, acetyl-CoA will enter the Krebs cycle. And we won't go into Krebs cycle during these videos. Later on we will probably. But for now we will stop there, and next we will review gluconeogenesis as well as glycogen synthesis. Thank you for watching. Please comment, subscribe, like, and tell me if I'm talking too fast, please. Thank you.